The RNA strand continues to grow. An A is added whenever the DNA template has a T. But when the DNA molecule has an A, the messenger RNA molecule incorporates a U. Remember that in an RNA molecule, uracil replaces thymine. Now the newly constructed messenger RNA molecule contains all the information to build a new polypeptide chain. It also has some non-coding segments. These are removed by other enzymes before the mRNA leaves the nucleus. When the messenger RNA molecule is complete, it's released from the nucleus. The newly formed messenger RNA leaves the nucleus and binds to a ribosome. Recall that ribosomes that make proteins for use within the cell are located in the cytoplasm. Ribosomes that make proteins for export and for use in the cell membrane are found on the rough ER. A ribosome has two parts, a large and small subunit. Ribosomal subunits are made in the nucleolus and assembled in the cytoplasm. A ribosome attaches to the messenger RNA and begins to translate the coded instructions.
The genetic code is written in three-letter words called triplets. Each triplet is a sequence of 3 nucleotide bases along a DNA strand. Each triplet specifies one amino acid for the polypeptide chain.
If you read a completed messenger RNA strand, you'll know the exact number and arrangement of all the amino acids that make up the new polypeptide chain. Translation of the messenger RNA molecule starts at a special codon called the start codon. Transfer RNA molecules pick up amino acids in the cytoplasm and carry them to the ribosomes. An amino acid attaches to one end of the transfer RNA. A sequence of 3 nucleotide bases known as an anticodon is at the opposite end of each transfer RNA molecule. Many types of transfer RNA molecules exist, and each has a different anticodon to carry one of the 20 different amino acids needed for protein construction. When a transfer RNA molecule approaches the ribosome, it follows the base pairing rule. The anticodon pairs with its complementary codon on the messenger RNA blueprint.
A ribosome bound to a messenger RNA strand has 2 binding sites for transfer RNA molecules. One site holds a tRNA attached to the growing polypeptide chain, while the other receives a tRNA carrying the next amino acid to be added to the chain. The anticodon of an incoming transfer RNA molecule binds to the corresponding messenger RNA codon.
The anticodon of an incoming transfer RNA molecule, which carries its amino acid, binds to the messenger RNA molecule codon at the A site of the ribosome. The ribosome moves along and reads each messenger RNA codon. As it's reading, it attaches a new amino acid to the polypeptide chain. This phase of translation is called elongation. Elongation adds amino acids to the polypeptide chain until a stop codon is reached. This ends translation and the chain is released. So, the next time you hear DNA mentioned in the news, remember, it's a library that has the necessary blueprints to manufacture the thousands of proteins in our bodies. Hi, I'm Suzanne Savoy, and I'll be your host for the video tutor discussion of muscle function. During exercise, we use hundreds of muscles. To understand how muscle contraction and coordination occur, we need to take a close look at the structure of skeletal muscle. When you're swimming, the muscles in your thighs are hard at work. Large skeletal muscles have thousands of muscle fibers bundled together within a connective tissue sheath. These bundles, called fascicles, contain blood vessels and nerves. Blood is needed to supply each muscle fiber with proper nutrients and oxygen during contraction and to carry away waste products generated. The nerves control the muscle contraction and monitor the position and length of the muscle. Each muscle fiber is a huge cell with hundreds of nuclei. The cell membrane of a muscle cell is called the sarcolemma. The myofibrils are attached to the sarcolemma at each end of the cell, so when they contract, the entire cell contracts with them. From the sarcolemma, small projections called T-tubules extend down into the interior of the cell. Inside the sarcoplasm, the T-tubules encircle each myofibril and contact regions of modified endoplasmic reticulum, called the sarcoplasmic reticulum. The sarcoplasmic reticulum contains cisternae filled with calcium ions. Within each myofibril, bundles of parallel thick and thin filaments are organized into repeating functional units called sarcomeres. Sarcomeres are the smallest functional units of the muscle fiber and are connected at junctions called Z-lines. Muscles contract because the thick and thin filaments of sarcomeres interact with each other. Each thin filament is composed of the protein actin. The globular actin molecules are joined together in a twisted strand. Thick filaments are composed of the protein myosin. Each myosin molecule is shaped like a double-headed golf club. The shafts are bundled together and the heads project outward in a spiral pattern. 6 actin filaments surround each myosin filament. The sliding filament theory explains the physical changes that occur in a sarcomere during a contraction. Our muscles contract when thin filaments slide along thick filaments. This pulls the two ends of the sarcomere, the Z-lines, closer together. You can understand this mechanism by using your hands. Hold out your hands so your palms are facing you and your fingertips are touching. Your wrists represent the Z-lines of the sarcomere boundaries, and your fingertips represent the thick and thin filaments. Slide the fingers of one hand between those of the other, and you see your wrists are closer together. Keeping your hands in the same position, look at this diagram of a relaxed sarcomere. Notice the distance between the two Z-lines. As the sarcomere contracts, the Z-lines move closer together. If you're understanding what we've seen so far, let's explore how the brain coordinates all these contracting muscles. Take this woman about to dive, for example. We all take it for granted that we know how to dive into a pool, but it involves a lot of communication between your muscular and nervous systems. Let's go back and watch her dive again. The coordination process starts with your brain sending signals to your skeletal muscles, telling them when to contract. Motor neurons in the CNS control skeletal muscle contractions. Each motor neuron can control several muscle fibers. The motor neuron and its dependent muscle fibers form a motor unit. When the neuron is active, all of the muscle fibers in the motor unit contract. The area where the terminal process of an axon communicates with the muscle cell is called a neuromuscular junction. The synaptic knob of the neuron and muscle fiber are separated by a small gap called the synaptic cleft. The neuron controls the muscle fiber by releasing a chemical neurotransmitter into the synaptic cleft. Let's follow the contraction process step by step. The first step is the arrival of an action potential at the neuromuscular junction. When the action potential gets to the neuromuscular junction, the synaptic knob releases molecules of the neurotransmitter acetylcholine into the synaptic cleft. Receptors on the sarcolemma bind the acetylcholine, and this triggers an action potential in the cell membrane of the muscle fiber. Remember, the sarcolemma forms T-tubules, which travel down toward the calcium cisternae in the sarcoplasmic reticulum. When the action potential passes the cisternae, calcium ions are released. These calcium ions flood the interior of the muscle fiber and the contraction cycle begins. A detailed look at a sarcomere shows that when your muscle is at rest,
Troponin holds tropomyosin against the actin strands, blocking the myosin binding sites.
but when calcium ions are released from cisternae in the sarcoplasmic reticulum, they attach to the troponin and the troponin rotates, moving the tropomyosin and exposing the myosin binding sites. This changes the position of the tropomyosin filaments so that myosin binding sites are exposed. The cross-bridge binding then occurs. A cross-bridge forms when the head of the myosin molecule attaches to a myosin binding site on the actin molecule. Next, the myosin heads pivot, pulling the actin filaments toward the center of the sarcomere. This sliding motion is dependent on the formation and pivoting of the cross bridges that link the actin and myosin filaments. The myosin head uses ATP energy to pull the actin filaments past each other toward the center of the sarcomere. What we've just learned is only one part of the big picture, because there are many cross bridges occurring at any given moment. Cross bridges break and reattach often as the thin filaments are pulled toward the center of the sarcomere. Each time the myosin head binds to a new site on the actin molecule, it shortens the sarcomere. In one muscle fiber contraction, each myosin head works its way across dozens of active sites along the actin molecule. When the muscle is fully contracted, the two Z lines are much closer together than when the sarcomere is relaxed. Each stroke requires a variety of complicated contraction and relaxation cycles. Muscle relaxation also involves several different steps.
When neural stimulation ends, the sarcoplasmic reticulum is able to lower the concentration of calcium ions in the sarcoplasm. The troponin and tropomyosin then return to their original positions.
This covers the active sites on the actin and prevents the myosin heads from forming cross bridges. Now the contraction is over. After viewing this material on skeletal muscle, you can now understand and appreciate the processes that take place in your muscles during swimming or simply walking down a flight of stairs. Hi, I'm Suzanne Savoy, and welcome to the video tutor discussion of the nervous system. This student is not only reading the computer screen, she's also listening, writing, even tapping her foot. She's also, even though you can't see these things, breathing, her heart's pumping, her body temperature is being regulated, and many more physiological functions are being simultaneously controlled and regulated by the brain. In order for all these functions to take place, specialized cells called neurons form a living network that senses changing conditions, processes sensory input, and then activates different body systems that carry out responses. All neurons have common features, but they still come in different shapes and sizes. A neuron has a large cell body containing the nucleus and the cytoplasmic organelles. Neurons also have small projections called dendrites.
Dendrites are short, highly branched processes
which receive signals from the environment and other neurons across synapses. Most neurons have a single, long axon, a cell process specialized for the propagation of action potentials. Axons are often wrapped in a thick, insulating layer called the myelin sheath. This speeds the rate of propagation. The tip of the axon is split into a cluster of fine telodendria. Each one is enlarged at the tip to form a synaptic knob. Within each synaptic knob are many synaptic vesicles containing neurotransmitter molecules. Neurotransmitters are chemicals that relay signals to other neurons or to effectors, such as muscle fibers or gland cells. Communication within the nervous system uses both electrical and chemical processes. When a neuron is in the resting state, a difference in the distribution of electrical charges is seen across the cell membrane. The exterior of the cell membrane has a positive charge, and the interior has a negative charge. Now the membrane is polarized. The separation of positive and negative charges by the cell membrane creates a potential difference called the resting membrane potential. These potential differences are measured in volts. The inside of the cell membrane has a slightly negative charge than the outside.
In a typical neuron, the resting membrane potential is minus 70 millivolts. Depolarization occurs when the membrane potential shifts towards 0 millivolts. An increase to minus 80 or minus 90 millivolts is called hyperpolarization.
A neuron may respond to any stimulus that changes the resting potential. The largest change in membrane potential is called an action potential. Action potentials occur in areas of excitable membrane. Two examples of excitable membranes are the sarcolemma of a skeletal muscle fiber and the axolemma of a neuron. An action potential is a sudden reversal in the polarity of a portion of the membrane. For a fraction of a second, the inside of that part of the membrane becomes positive compared to the outside.
Once the action potential is triggered, it is propagated along the membrane. The nervous system transmits information in the form of propagated action potentials.
Resting potentials and action potentials are caused by ion movement across the cell membrane. The concentration of sodium ions is greater outside the cell than inside the cell. The concentration of potassium ions is greater inside the cell than outside. This unequal distribution of ions is maintained by membrane proteins that function as channels or pumps.
As you learned during the segment on membrane transport, the cell membrane is selectively permeable.
Small ions like sodium and potassium can diffuse through channels in the cell membrane following electrical and chemical gradients.
But the suspended proteins and other large organic molecules are kept inside the cell.
Most of these large molecules are negatively charged. Two passive forces explain the ion movement across the membranes: chemical forces from existing concentration gradients and electrical forces from the attraction between positive and negative charges and the repulsion of two positive or two negative charges. Potassium ions are the most abundant cations in the cytosol. Potassium leak channels allow potassium ions to diffuse out following the concentration gradient.
But the loss of potassium ions is opposed by electrical forces.
The potassium ions have a positive charge, and the interior of the cell membrane is negatively charged.
If no other factors were involved, the net outward movement of potassium would stop at a transmembrane potential of around minus 90 millivolts.
At this point, the chemical gradient driving potassium out of the cell would be balanced by the electrical gradient attracting potassium into the cell.
Relatively